Welcome to Chapter 3, Section 4, Unique Characteristics of Eukaryotic Cells. Learning objectives in this section are: explain the distinguishing characteristics of eukaryotic cells, describe internal and external structures of prokaryotic cells in terms of their physical structure, chemical structure, and function, identify and describe structures and organelles unique to eukaryotic cells, compare and contrast similar structures found in prokaryotic and eukaryotic cells. Eukaryotic organisms include protozoans, algae, fungi, plants, and animals. Some eukaryotic cells are independent, single-celled microorganisms, whereas others are part of multicellular organisms. The cells of eukaryotic organisms have several distinguishing characteristics. Above all, eukaryotic cells are defined by the presence of a nucleus surrounded by a complex nuclear membrane. Also, eukaryotic cells are characterized by the presence of membrane-bound organelles in the cytoplasm. Organelles such as mitochondria, the Endoplasmic Reticulum, Golgi Apparatus, lysosomes, and peroxisomes are held in place by the cytoskeleton, an internal network that supports transport of intracellular components and helps maintain cell shape. The genome of eukaryotic cells is packaged in multiple rod-shaped chromosomes as opposed to the single circular-shaped chromosome that characterizes most prokaryotic cells. Now, this table compares the characteristics of eukaryotic cell structures with those of bacteria and archaea. First, we have the relative size of bacteria and archaea are similar to each other in the single-digit micrometer range, whereas eukaryotes range from about 5 to 20 micrometers. That makes it look like molar using the capital M. That should be a lowercase m. Surface to area volume ratio is high in bacteria and archaea, but low in eukaryotes. And neither bacteria nor archaea possess a nucleus, whereas eukaryotes do. More characteristics. Bacterial chromosomes tend to be single, circular, haploid, and lack histones. Archaeal cells share those characters, except some of them do contain histone-like proteins. Eukaryotes have multiple chromosomes that are linear. They can be haploid or diploid, or they can alternate between the two, and they do contain histones. Both bacteria and archaea engage in binary fission, whereas eukaryotes engage in mitosis and meiosis. Membrane lipid composition of bacteria are ester-linked straight-chain fatty acids and occur in a bilayer, which those characters they share with eukaryotes. Eukaryotes additionally tend to have sterols in the membrane. Meanwhile, archaeal cells are ether-linked. They use branched isoprenoids, and they occur in bilayers or monolayers. Bacterial cell walls are composed of peptidoglycan or have no cell wall. The cell walls of archaea can be pseudopeptidoglycan or glycopeptide or polysaccharide or protein, the S layer, or none. The cell walls of eukaryotes, none can be an option, and they are for most others, but they can be composed of cellulose, which include plants and some algae, chitin, mollusks, insects, crustaceans, and fungi, silica for some algae, like diatoms. Now, for motility structures, bacteria and archaea have rigid spiral flagella that have either flagellin or archaeal flagellins, whereas the flagella in eukaryotes tend to be flexible. Instead of spinning like the others do, they can wave back and forth. Eukaryotes can also have cilia composed of microtubules that wave back and forth to impart motion. Neither bacteria nor archaea have membrane-bound organelles, but eukaryotes do. There is no endomembrane system in either bacteria or archaea. The eukaryotes do have that, and that includes the Endoplasmic Reticulum, the Golgi Apparatus, and Lysosomes. The ribosomes are 70 Svedberg units in archaea and bacteria, while in the eukaryotes there is an 80S in the cytoplasm and attached to the rough endoplasmic reticulum, and the 70S in mitochondria and chloroplasts, being as those were derived from bacteria. Cell morphologies. Eukaryotic cells display a wide variety of different cell morphologies. Possible shapes include spheroid, ovoid, cuboidal, cylindrical, flat, lenticular, fusiform, discoidal, crescent, ring stellate, and polygonal. Some eukaryotic cells are irregular in shape, and some are capable of changing shape. The shape of a particular type of eukaryotic cell may be influenced by factors such as its primary function, the organization of its cytoskeleton, the viscosity of its cytoplasm, the rigidity of its cell membrane or cell wall, if it has one, and the physical pressure exerted on it by the surrounding environment and/or adjoining cells. Unlike prokaryotic cells, in which DNA is loosely contained in the nucleoid region, eukaryotic cells possess a nucleus, which is surrounded by a complex nuclear membrane that houses the DNA genome. By containing the cell's DNA, the nucleus ultimately controls all activities of the cell and also serves an essential role in reproduction and heredity. Eukaryotic cells typically have their DNA organized into multiple linear chromosomes. The DNA within the nucleus is highly organized and condensed to fit inside the nucleus, which is accomplished by wrapping the DNA around proteins called histones. Although most eukaryotic cells have only one nucleus, exceptions exist. For example, protozoans of the genus Paramecium typically have two complete nuclei, a small nucleus that is used for reproduction, the micronucleus, and a large nucleus that directs cellular metabolism, the macronucleus. Additionally, some fungi transiently form cells with two nuclei, called heterokaryotic cells, during sexual reproduction. Cells whose nuclei divide, but whose cytoplasm does not, are called choanocytes. The nucleus is bound by a complex nuclear membrane, often called the nuclear envelope, that consists of two distinct lipid bilayers that are contiguous with each other. Despite these connections between the inner and outer membranes, each membrane contains unique lipids and proteins on its inner and outer surfaces. The nuclear envelope contains nuclear pores, which are large rosette-shaped protein complexes that control the movement of materials into and out of the nucleus. The overall shape of the nucleus is determined by the nuclear lamina, a meshwork of intermediate filaments found just inside the nuclear envelope membranes. Outside the nucleus, additional intermediate filaments form a looser mesh and serve to anchor the nucleus in position within the cell. Nucleolus. The nucleolus is a dense region within the nucleus where ribosomal RNA, rRNA, biosynthesis occurs. In addition, the nucleolus is the site where assembly of ribosomes begins. Pre-ribosomal complexes are assembled from ribosomal RNA and proteins in the nucleolus. They are then transported out to the cytoplasm, where ribosome assembly is complete. Ribosomes. Ribosomes found in eukaryotic organelles such as mitochondria or chloroplasts have 70S ribosomes, the same size as prokaryotic ribosomes. However, non-organelle-associated ribosomes in eukaryotic cells are 80S ribosomes, composed of a 40S small subunit and a 60S large subunit. In terms of size and composition, this makes them distinct from the ribosomes of prokaryotic cells. The two types of non-organelle-associated eukaryotic ribosomes are defined by their location in the cell: free ribosomes and membrane-bound ribosomes. Free ribosomes are found in the cytoplasm and serve to synthesize water-soluble proteins. Membrane-bound ribosomes are found attached to the rough endoplasmic reticulum and make proteins for insertion into cell membrane or proteins destined for export from the cell. There are not ribosomes that are attached permanently to the rough ER. They assemble as needed and then bind based on localization signals that are on the message itself. Endomembrane system. The endomembrane system, unique to eukaryotic cells, is a series of membranous tubules, sacs, and flattened discs that synthesize many cell components and move materials around within the cell. Because of their larger cell size, eukaryotic cells require this system to transport materials that cannot be dispersed by diffusion alone. The endomembrane system comprises several organelles and connections between them, including the endoplasmic reticulum, Golgi apparatus, lysosomes, and vesicles. Endoplasmic reticulum. The endoplasmic reticulum, ER, is an interconnected array of tubules and cisternae, flattened sacs, with a single lipid bilayer. The space inside of the cisternae are called the lumen of the ER. There are two types of ER, rough endoplasmic reticulum, RER, and smooth endoplasmic reticulum, SER. The two different types of ER are sites for the synthesis of distinctly different types of molecules. RER is studded with ribosomes bound on the cytoplasmic side of the membrane. These ribosomes make proteins destined for the plasma membrane. Following synthesis, these proteins are inserted into the membrane of the RER. Small sacs of the RER containing these newly synthesized proteins then bud off as transport vesicles and move either to the Golgi apparatus for further processing, directed to the plasma membrane, to the membrane of another organelle, or out of the cell. Transport vesicles are single lipid, bilayer, membranous spheres with hollow interiors that carry molecules. SER does not have ribosomes and therefore appears smooth. It is involved in the biosynthesis of lipids, carbohydrate metabolism, and detoxification of toxic compounds within the cell. The Golgi apparatus was discovered within the endomembrane system in 1898 by Italian scientist Camillo Golgi, who lived from 1843 to 1926, who developed a novel staining technique that showed stacked membrane structures within the cells of Plasmodium, the causative agent of malaria. The Golgi apparatus is composed of a series of membranous discs called dictyosomes, each having a single lipid bilayer that are stacked together. Enzymes in the Golgi apparatus modify lipids and proteins transported from the ER to the Golgi, often adding carbohydrate components to them, producing glycolipids, glycoproteins, or proteoglycans. Glycolipids and glycoproteins are often inserted into the plasma membrane and are important for signal recognition by other cells or infectious particles. Different types of cells can be distinguished from one another by the structure and arrangement of the glycolipids and glycoproteins contained in their plasma membranes. These glycolipids and glycoproteins commonly also serve as cell surface receptors. Transport vesicles leaving the ER fuse with a Golgi apparatus on its receiving or cis face. The proteins are processed within the Golgi apparatus, and then additional transport vesicles containing the modified proteins and lipids pinch off from the Golgi apparatus on its outgoing or trans face. These outgoing vesicles move to and fuse with the plasma membrane or the membrane of other organelles. Exocytosis is the process by which secretory vesicles, spherical membranous sacs, release their contents to the cell's exterior. All cells have constitutive secretory pathways in which secretory vesicles transport soluble proteins that are released from the cell continually, constitutively. Certain specialized cells also have regulated secretory pathways which are used to store soluble proteins in secretory vesicles. Regulated secretion involves substances that are only released in response to certain events or signals. For example, certain cells of the human immune system, mast cells for example, secrete histamine in response to the presence of foreign objects or pathogens in the body. Histamine is a compound that triggers various mechanisms used by the immune system to eliminate pathogens. Lysosomes. In the 1960s, Belgian scientist Christian de Duve, 1917 to 2013, discovered lysosomes, membrane-bound organelles of the endomembrane system that contain digestive enzymes. Certain types of eukaryotic cells use lysosomes to break down various particles, such as food, damaged organelles or cellular debris, microorganisms, or immune complexes. Compartmentalization of the digestive enzymes within the lysosome allow the cell to efficiently digest matter without harming the cytoplasmic components of the cell. Peroxisomes. Christian de Duve is also credited with the discovery of peroxisomes, membrane-bound organelles that are not part of the endomembrane system. Peroxisomes form independently in the cytoplasm from the synthesis of peroxin proteins by free ribosomes and the incorporation of these peroxin proteins into existing peroxisomes. Growing peroxisomes then divide by a process similar to binary fission. Peroxisomes were first named for their ability to produce hydrogen peroxide, a highly reactive molecule that helps to break down molecules such as uric acid, amino acids, and fatty acids. Peroxisomes also possess the enzyme catalase, which can degrade hydrogen peroxide. Along with the SER, peroxisomes also play a role in lipid biosynthesis. Like lysosomes, the compartmentalization of these degradative molecules within an organelle helps protect the cytoplasmic contents from unwanted damage. The peroxisomes of certain organisms are specialized to meet their particular functional needs. For example, glyoxysomes are modified peroxisomes of yeast and plant cells that perform several metabolic functions, including the production of sugar molecules. Similarly, glycosomes are modified peroxisomes made by certain trypanosomes, the pathogenic protozoans that cause Chagas disease and African sleeping sickness. Cytoskeleton. Eukaryotic cells have an internal cytoskeleton made of microfilaments, intermediate filaments, and microtubules. This matrix of fibers and tubes provides structural support as well as a network over which materials can be transported within the cell and on which organelles can be anchored. For example, the process of exocytosis involves the movement of a vesicle via the cytoskeletal network to the plasma membrane, where it can release its contents. Microfilaments are composed of two intertwined strands of actin, each composed of actin monomers forming filamentous cables 6 nanometers in diameter. The actin filaments work together with motor proteins like myosin to effect muscle contractions in animals or the amoeboid movement of some eukaryotic microbes. In amoeboid organisms, actin can be found in two forms: a stiffer polymerized gel form and a more fluid unpolymerized soluble form. Actin in the gel form creates stability in the ectoplasm, the gel-like area of cytoplasm just inside the plasma membrane of amoeboid protozoans. Temporary extensions of the cytoplasmic membrane called pseudopodia, meaning false feet, are produced through the forward flow of soluble actin filaments into the pseudopodia, followed by the gel-sol cycling of the actin filaments, resulting in cell motility. Once the cytoplasm extends outward, forming a pseudopodium, the remaining cytoplasm flows up to join the leading edge, thereby creating forward locomotion. Beyond amoeboid movement, microfilaments are also involved in a variety of other processes in eukaryotic cells, including cytoplasmic streaming, the movement or circulation of cytoplasm within the cell, cleavage furrow formation during cell division, and muscle movement in animals. These functions are the result of the dynamic nature of microfilaments, which can polymerize and depolymerize relatively easily in response to cellular signals, and their interactions with molecular motors in different types of eukaryotic cells. Intermediate filaments. Intermediate filaments are a diverse group of cytoskeletal filaments that act as cables within the cell. They are termed intermediate because their 10 nanometer diameter is thicker than that of actin, but thinner than that of microtubules. They are composed of several strands of polymerized subunits that, in turn, are made up of a wide variety of monomers. Intermediate filaments tend to be more permanent in the cell and maintain the position of the nucleus. They also form the nuclear lamina, lining or layer, just inside the nuclear envelope. Additionally, intermediate filaments play a role in anchoring cells together in animal tissues. The intermediate filament protein, desmin, is found in desmosomes, the protein structures that join muscle cells together and help them resist external physical forces. The intermediate filament protein, keratin, is a structural protein found in hair, skin, and nails. Microtubules are a third type of cytoskeletal fiber composed of tubulin dimers, alpha tubulin and beta tubulin. They form hollow tubes 23 nanometers in diameter that are used as girders within the cytoskeleton. Like microfilaments, microtubules are dynamic and have the ability to rapidly assemble and disassemble. Microtubules also work with motor proteins, such as dynin and kinesin, to move organelles and vesicles around within the cytoplasm. Additionally, microtubules are the main components of eukaryotic flagella and cilia, composing both the filament and the basal body components. In addition, microtubules are involved in cell division, forming the mitotic spindle that serve to separate chromosomes during mitosis and meiosis. The mitotic spindle is produced by two centrosomes, which are essentially microtubule organizing centers at opposite ends of the cell. Each centrosome is composed of a pair of centrioles positioned at right angles to each other, and each centriole is an array of 9 parallel microtubules arranged in triplets. Mitochondria. The large complex organelles in which aerobic cellular respiration occurs in eukaryotic cells are called mitochondria. The term mitochondrion was first coined by German microbiologist Carl Benda in 1898 and was later connected with the process of respiration by Otto Warburg in 1913. Scientists during the 1960s discovered that mitochondria have their own genome and 70S ribosomes. The mitochondrial genome was found to be bacterial when it was sequenced in 1976. These findings ultimately supported the endosymbiotic theory proposed by Lynn Margulis, which states that mitochondria originally arose through an endosymbiotic event in which a bacterium capable of aerobic cellular respiration was taken up by phagocytosis into a host cell and remained as a viable intracellular component. Each mitochondrion has 2 lipid membranes. The outer membrane is a remnant of the original host cell's membrane structure. The inner membrane was derived from the bacterial plasma membrane. The electron transport chain for aerobic respiration uses integral proteins embedded in the inner membrane. The mitochondrial matrix, corresponding to the location of the original bacterium's cytoplasm, is the current location of many metabolic enzymes. It also contains mitochondrial DNA and 70S ribosomes. Invaginations of the inner membrane, called cristae, evolved to increase surface area for the location of biochemical reactions. The folding patterns of cristae differ among various types of eukaryotic cells and are used to distinguish different eukaryotic organisms from each other. One of the more recent ideas about these folding points is that the ATP synthases occur in pairs that are angled next to each other. Chloroplasts. Plant cells and algal cells contain chloroplasts, the organelles in which photosynthesis occurs. All chloroplasts have at least 3 membrane systems: the outer membrane, the inner membrane, and the thylakoid membrane system. Inside the outer and inner membranes is the chloroplast stroma, a gel-like fluid that makes up much of a chloroplast's volume and in which the thylakoid system floats. The thylakoid system is a highly dynamic collection of folded membrane sacs. It is where the green photosynthetic pigment chlorophyll is found and the light reactions of photosynthesis occur. In most plant chloroplasts, the thylakoids are arranged in stacks called grana, singular granum, whereas in some algal chloroplasts, the thylakoids are free-floating. Other organelles similar to mitochondria have arisen in other types of eukaryotes, but their roles differ. Hydrogenosomes are found in some anaerobic eukaryotes and serve as the location of anaerobic hydrogen production. Hydrogenosomes typically lack their own DNA and ribosomes. Kinetoplasts are a variation of the mitochondria found in some eukaryotic pathogens. In these organisms, each cell has a single long-branched mitochondrion in which kinetoplast DNA, organized as multiple circular pieces of DNA, is found concentrated at one pole of the cell. Microconnections: mitochondria-related organelles in protozoan parasites. Many protozoans, including several protozoan parasites that cause infections in humans, can be identified by their unusual appearance. Distinguishing features may include complex cell morphologies, the presence of unique organelles, or the absence of common organelles. The protozoan parasite Giardia lamblia and Trichomonas vaginalis are 2 examples. Giardia lamblia, a frequent cause of diarrhea in humans and many other animals, is an anaerobic parasite that possesses 2 nuclei and several flagella. Its Golgi apparatus and endoplasmic reticulum are greatly reduced, and it lacks mitochondria completely. However, it does have organelles known as mitosomes, double membrane-bound organelles that appear to be severely reduced mitochondria. This has led scientists to believe that Giardia lamblia's ancestors once possessed mitochondria that evolved to become mitosomes. Trichomonas vaginalis, which causes the sexually transmitted infection vaginitis, is another protozoan parasite that lacks conventional mitochondria. Instead, it possesses hydrogenosomes, mitochondrial-related double membrane-bound organelles that produce molecular hydrogen used in cellular metabolism. Scientists believe that hydrogenosomes, like mitosomes, also evolved from mitochondria. Plasma membrane. The plasma membrane of eukaryotic cells is similar in structure to the prokaryotic plasma membrane in that it is composed mainly of phospholipids forming a bilayer with embedded peripheral and integral proteins. These membrane components move within the plane of the membrane according to the fluid mosaic model. However, unlike the prokaryotic membrane, eukaryotic membranes contain sterols, including cholesterol, that alter membrane fluidity. Additionally, many eukaryotic cells contain some specialized lipids, including sphingolipids, which are thought to play a role in maintaining membrane stability, as well as being involved in signal transduction pathways and cell-to-cell communication. Membrane transport mechanisms. The process of simple diffusion, facilitated diffusion, and active transport are used in both eukaryotic and prokaryotic cells. However, eukaryotic cells also have the unique ability to perform various types of endocytosis, the uptake of matter through plasma membrane invagination and vacuole vesicle formation. A type of endocytosis involving the engulfment of large particles through membrane invagination is called phagocytosis, which means cell eating. In phagocytosis, particles, or other cells, are enclosed in a pocket within the membrane, which then pinches off from the membrane to form a vacuole that completely surrounds the particle, or other cells. Another type of endocytosis is called pinocytosis, which means cell drinking. In pinocytosis, small dissolved materials and liquids are taken into the cell through small vesicles. Saprophytic fungi, for example, attain their nutrients from dead and decaying matter largely through pinocytosis. Receptor-mediated endocytosis is a type of endocytosis that is initiated by specific molecules called ligands when they bind to cell surface receptors on the membrane. Receptor-mediated endocytosis is the mechanism that peptide and amine-derived hormones use to enter cells, and is also used by various viruses and bacteria for entry into host cells. The process by which secretory vesicles release their contents to the cell's exterior is called exocytosis. Vesicles move towards the plasma membrane and then meld with the membrane, ejecting their contents out of the cell. Exocytosis is used by cells to remove waste products and may also be used to release chemical signals that can be taken up by other cells. Cell wall. In addition to a plasma membrane, some eukaryotic cells have a cell wall. Cells of fungi, algae, plants, and even some protists have cell walls. Depending upon the type of eukaryotic cell, cell walls can be made from a wide variety of materials, including cellulose, fungi and plants, biogenic silica, calcium carbonate, agar, and carrageenan, protists and algae, or chitin, fungi. In general, all cell walls provide structural stability for the cell and protection from environmental stresses such as desiccation, changes in osmotic pressure, and traumatic injury. Extracellular matrix. Cells of animals and some protozoans do not have cell walls to help maintain shape and provide structural stability. Instead, these type of eukaryotic cells produce an extracellular matrix, or ECM, for this purpose. They secrete a sticky mass of carbohydrates and proteins into the spaces between adjacent cells. Some protein components assemble into a basement membrane to which the remaining ECM components adhere. Proteoglycans typically form the bulky mass of the extracellular matrix, while fibrous proteins, like collagen, provide strength. Both proteoglycans and collagen are attached to fibronectin proteins, which in turn are attached to integrin proteins. These integrin proteins interact with transmembrane proteins in the plasma membranes of eukaryotic cells that lack cell walls. In animal cells, the ECM allows cells within tissues to withstand external stresses and transmit signals from the outside of the cell to the inside. The amount of extracellular matrix is quite extensive in various types of connective tissues, and variations in the extracellular matrix can give different types of tissues their distinctive properties. In addition, a host cell's extracellular matrix is often the site where microbial pathogens attach themselves to establish infection. For example, Streptococcus pyogenes, the bacterium that causes strep throat and various other infections, binds to fibronectin in the ECM of the cells lining the oropharynx, the upper region of the throat. Flagella and cilia. Some eukaryotic cells use flagella for locomotion. However, eukaryotic flagella are structurally distinct from those found in prokaryotic cells. Whereas the prokaryotic flagellum is a stiff, rotating structure, a eukaryotic flagellum is more like a flexible whip composed of 9 parallel pairs of microtubules surrounding a central pair of microtubules. This arrangement is referred to as a 9+2 array. The parallel microtubes use dynein motor proteins to move relative to each other, causing the flagellum to bend. Cilia, singular cilium, are a similar external structure found in some eukaryotic cells. Unique to eukaryotes, cilia are shorter than flagella and often cover the entire surface of a cell. However, they are structurally similar to flagella, a 9+2 array of microtubules, and use the same mechanism for movement. A structure called a basal body is found at the base of each cilium and flagellum. The basal body, which attaches the cilium or flagellum to the cell, is composed of an array of triplet microtubules similar to that of a centriole, but embedded in the plasma membrane. Because of their shorter length, cilia use a rapid, flexible waving motion. In addition to motility, cilia may have other functions, such as sweeping particles past or into cells. For example, ciliated protozoans use the sweeping of cilia to move food particles into their mouth parts, and ciliated cells in the mammalian respiratory tract beat in synchrony to sweep mucus and debris up and out of the lungs, the mucilatory escalator. Clinical focus: resolution. Since the amoxicillin has not resolved Barbara's case of pneumonia, the PA prescribes another antibiotic, azithromycin, which targets bacterial ribosomes rather than peptidoglycan. After taking the azithromycin as directed, Barbara's symptoms resolve and she finally begins to feel like herself again. Presuming no drug resistance to amoxicillin was involved, and given the effectiveness of the azithromycin, the causative agent of Barbara's pneumonia is most likely Mycoplasma pneumonia. Even though this bacterium is a prokaryotic cell, it is not inhibited by amoxicillin because it does not have a cell wall and therefore does not make peptidoglycan. And that brings us to the end of Chapter 3. Join me next time for Chapter 4, Prokaryotic Diversity. All right, till then.